Great. So just to kind of introduce what I'll be presenting today, we developed the cross-trial, cross-study capability in conjunction with a project at the FDA last year. And up until now, if you wanted to do studies with patients from multiple studies at once, you would have to do them individually in tranSMART. And what we've enabled is the ability to do analyses across multiple trials at once. In the FDA's use case, they were looking at device data, drug data, and vaccine data, and they wanted to determine if there were specific treatment options that were more effective than others, if there were more adverse events, and different safety outcomes across multiple clinical trials, so they can identify potentially a treatment, being a drug, device, or vaccine, that may be more effective than others in certain scenarios. So in developing this work, we had our team at Converge Health that worked on this, and then we worked at the various centers at the FDA, including CDRH, CBER, and CDER, which are the three primary centers for device studies, vaccine studies, and drug studies. And with that, I'll just move right into the demo of what I'll be showing today. And just to give you a quick introduction to that as well, I'll be showing a system that's not version 1.2, which shows the full functionality of how this cross-study will work, and then I'll show it again in 1.2. The reason for that is I just don't have enough studies in 1.2 on the instance that we have available to show the full functionality there. So I'm actually going to start with a different instance here, and the first thing what I'll do is I'm in Dataset Explorer, which is now called Analyze, and I'll go into the Across Trials section, and in this case what I might want to do is, my example is comparing asthma and COPD patients. So I can just drag in asthma and drag in COPD patients and look at summary statistics across both these groups. And I can see all the same types of information you would if you were looking at one specific study, which is the histogram of age. You see the sex information, and if race information was available for these clinical trials, that would be shown here as well. I can also drag in a node just as I would with a single study. I can drag that in. And I can see now for the smoking behavior, I can see ex-smokers, non-smokers, and smokers in both of these subsets. And to give you an idea of what I'm looking at in this system, I can look at the studies, and I can see I have 2 asthma studies. I have a bunch of breast cancer studies, but that's not going to be applicable to these particular conditions I'm looking at. And then I have COPD studies. So what this is doing is comparing the 2 sets of patients for the asthma and the 2 sets of patients for the COPD studies. Now if I want to take this one step further, what I can do is do like a box plot and determine if there's maybe a lab value or some sort of marker that's impacted by their condition or by the different elements included in the study. So in this case, when I look here, I see that roughly the patients who are 45 and older fall into both groups. The COPD patients are a little older. So what I'm going to do is pick a population now that's just folks who are over 45 across both sets here. And that way I'm getting a similar set of population. And then I'll go into the advanced workflow and do a box plot. And now what I'm going to do is look at the different trial arms. So I can drag in the different trial arms here and compare folks like healthy controls to severe asthma patients. And I want to look and see if there's a lab value or some sort of finding that might be affected by their diagnosis or their condition in the study. For asthma and COPD, forced expiratory volume is a reasonable thing to look at. So when I run this, it's going to come back and it's going to give me a standard box plot of all these different traits here across the board here. And as you can see, as you might expect, I can see that I've got healthy controls, I've got severe asthma, and I've got a broad range of difference across these here. So I can see a whole lot of what's going on. And right now I think I'm actually just comparing the 2 asthma studies. To take that a step further, I can drag in the COPD studies here as well. And then what I can do is enable binning on the independent variable, which is a categorical variable. I'll do manual binning. And now what I can do is look at, say, the healthy controls in one group, who are the folks that don't have COPD or are listed as healthy controls. The COPD, I'm expecting to probably have the worst respiratory volume there, so I'm going to put them down here. I've got my severe asthma, and then most of these are non-severe asthmas. So now what I'm essentially doing is looking across these 4 studies at all the patients and just binning them into groups here, running the same box plot. And now what will come back is 4 different box plots, one for each of those categories. And as you can see, a trend develops here based on their condition. So that's the primary functionality. I'll quickly show in 1.2 how this works as well. Very similar. So here I've got the navigate terms, the across trials piece here. And just because I have a little bit less data, I'll look at this a little differently. And I'm going to look straight at the forced expiratory volume. And what I'm going to do is subset this by their actual volume here, what their value was. I think 80 was a number close to the middle, so I'm going to look at less than 80 in one subset and greater than or equal to 80 in the other subset. And when I run summary statistics, I can get the same type of histograms, the same information. Again, no race data was collected for these particular patients. And then if I move on to the advanced workflow, what I'll do is I'll clear this analysis. I'll look at maybe one particular study here. I could drag in another if there was data for it, and then move to the advanced workflow, run the same box plot, and I can do the same type of thing here. So I'll look at the trial arms. I can drag in the healthy controls and the non-severe asthmas, whatever they might be, look at that against forced expiratory volume and run this. And I'll get a similar type of graphic back. Again, the data is just reflective of really one study in this particular case, so I'm not looking at multiple studies right now. But the functionality is all there to support the exact same use case. So, are there any questions on anything I just shown, or? Okay. So I think that's all I had. So with that, I can move on and hand control over. I think Jay was next. Is that right?